Well, maybe I can assume that everything was going okay technically, but I still always ask the question at the end of the first half. My usual question: did everybody hear clearly and continuously in the first half? I didn't get any error messages here, so I'm assuming you did. And I'm hoping somebody will say yes. Hello. I think I saw. Oh, good. We see yeses. And I have complete freedom in what I do here, and sometimes the things that I do here make certain people angry. Like my medical school. But I have complete freedom. Well, I'm very, very thankful for my medical school to pay me. So while I teach the students in the classroom, everybody out there in Buzzardville also benefits. And some of the medical schools also use my lectures. But the other two people I want to thank are Matt and Jim. You see, we said thanks. That's something I just realized. You should be thankful for the people that help you and contribute to this. Matt is the person whose office that I share. He's a very, very prominent attorney, and he has been gracious enough to let me use one of his rooms for my broadcasting studio. In fact, yesterday he even helped me improve the quality here. He got me a nice light so my face doesn't look so dark, and we got a nice backdrop. And of course, probably the person who has been helping me the most for 10 years now has been Jim. Jim is my buddy. Jim took me to China. Jim, now we're talking about genetics today. I should give thanks to Jim because Jim has supported these webinars with the webinar fees and charges for many, many, many years. So thank you, San Juan Bautista. Thank you, Matt. And thank you, Jim. Let's continue just like we threatened we were going to do and get into the second half. We've done more than half already. So let's start talking about another group, another classical genetics disease. Technically, it's not a lysosomal storage disease, but it's called alkaptonuria. It's kind of a funny name because alkap, ALC, is basically the an Arabic word for alkali. It's where the word alkali comes from. And apto or capto is actually Greek, and it means black, okay? So why would the word alkaptonuria mean black alkali? Because this is a disease which results often in black urine. And when the urine is exposed to air, it becomes even blacker. So it means an alkali that is, when it's oxygenated, becomes blacker. That's because of its problem with the degradation and processing of the amino acids phenylalanine and tyrosine. If you remember, tyrosine is very important in the production of pigmented materials as well, like melanin. So the urine is black, the nails are black, the skin is black, the joint cartilage is black. It could be so black that sometimes you could see it through the skin. And it's a very, very simple concept. If you have a compound called homogentisic acid, okay, and you have a deficiency of homogentisic acid oxidase, it's not degraded, it's not oxygenated, and it's in the process of basically in the tyrosine and phenylalanine metabolic pathways, you now have buildup of homogentisic acid. It gets to the joints, it gets to the skin, it gets to the nails, and it goes abundantly in the urine. It's already probably black, but when it's finally exposed to the air in the urine, it gets blacker. So that's how you get the name alkaptonuria. And here we have an example of urine. Now it's black. And look, this is an ear cartilage. Remember, the ear has elastic cartilage, but even through the skin, you could see a blackness through there. And here you could see some blackening of the nails and perhaps the skin underneath them as well. So this is a classical genetic disease. And look, here's a part of the skin now. And you could tell this is epidermis, and you could tell most of this is connective tissue is the dermis. But look, we got buildup of material in the dermis now, which is black. let's say, tyrosine abnormal byproducts, which is causing the skin to be black as well. So that was probably one of the first classical genetic diseases to be worked on, and it still can occur. Probably another really classical genetics disease, okay, is the neurofibromatosis. Now, I know you probably have heard that neurofibromatosis results in bumps, little fibromas, and little pigmented spots called cafe au lait, because they just happen to be the same exact color as my coffee. And here are some bumps on the knees. And look, there's even in the iris here, a little pigmented area, okay? What do these three cells all have in common? Well, they're all derived from neural crest, and they differentiate into cells which surround nerves, or melanocytes, or more typically the endoneurial fibroblasts. So if you have a genetic problem interfering, which basically is causing a overexpression of these, you now have the disease, or classically neurofibromatosis, and very classically neurofibromatosis 1, which is called also von Recklinghausen disease. There's another variation of it, neurofibromatosis 2, in which you very often can have buildup of little neural tumors, like acoustic neuromas, remember 6 nerve tumors at the cerebellopontine angle, as well as meningiomas as well. So let's get a better clinical look at this. Here is neurofibromatosis 2. What are these white lumps bilateral at the cerebellar pontine angle? Well, they're tumors, and they're bilateral tumors. So I think this is almost a knee-jerk reflex that if you ever do get a patient with head problems, hearing problems, and you get a CT and they say that there's tumors at both, cerebellar pontine angles, you could think this person very well may have neurofibromatosis, number 2 especially. Now take a look at this brain on a sagittal view. You could see multiple tumors, and it looks like the tumors may not be completely in the brain, but perhaps growing into the brain from the dura. Well, these are meningiomas. So just as though you might suspect neurofibromatosis 2 with bilateral or multiple acoustic neuromas, you can expect also the same disease if there are multiple meningiomas as well. Okay, this is an interesting concept, multifactorial inheritance. Remember, we also said that this could be called multigenic, and both of them are not really the best terms in the world. The term that I kind of invented, because for me, it makes it better to understand, is that your protoplasm, your flesh, your body, is basically gotten from your family. So you have the same type of cells, genes, susceptibility to disease as your family, even if you can't narrow it down to one single gene or 2 single genes, or the so-called environmental effect as well. And diseases of multifactorial inheritance, I guess if you wanted to say every disease could be, that wouldn't be entirely false, but classical features like hair color, eye color, skin color, height, intelligence, and even diabetes, type 2, which is even more complicated. What do these all things have in common? Okay, is they are not either the presence or absence of some things. They are actually regarded as being different. Like you could have a wide variety of intelligence, of height, of skin color. So it's not a case of being present or not being present. It's a disease of a quantitation rather than when it's on or off. Okay. So we are now moving from the discussion of one gene into multi-genes. And if you wanted to say that just about everything in a way could be related to multi-genes, you can. Let's see if we have anything more to say about that. Yeah. Uh, we say it's a number of genes, and the word that I was using before were continuous traits rather than discontinuous traits. That's the word I was struggling with. In other words, a disease of continuous something is something that has a spectrum, big, middle, small. Whereas something that's a discontinuous trait is something that's either present or absent, okay, like diabetes 1, for example. It's determined by a number of genes, and even though you may not be able to pinpoint those genes, or even if you did, there are environmental factors. Let's just say you probably have a disease of multifactorial inheritance if there's a greater than a 5% chance of a first-degree relative having it, like a son or a mother or a father or a daughter, okay? If the normal incidence of a disease is, 1/10 of a percent or something, or 1/100th of it, and all of a sudden you've got more than a 5% chance, it may be multigenic or multifactorial. So I know what you're thinking. You're thinking, well, if you're an identical twin, then it's probably going to be a hell of a lot more likely that your other identical twin is going to have it. Well, it's way more than 5%, but I got news for you. It's less than 100 as well. And I'm not going to give you an exact percentage because I don't think that's ever been agreed upon, because I don't think even that the multifactorial diseases have been all agreed upon. So this 5% possibility in first-degree relatives is more than 5% if multiple children have it, okay? So this sounds like a very vague sort of type of disease, but don't think of it as, the search to try to find another gene. Just think of it as something as a soil theory. Your closer relatives have closer genetics than further relatives. Look at this list. And I think I got this from Robbins. These are generally regarded as genetic diseases, but they cannot be traced, to one single gene. And look at some of these genes, for example, like hypertension. My God, half of everybody that walks into an internist's office probably has hypertension, okay? But cleft lip, congenital heart diseases, these are multifactorial, cannot be traced down to a single gene. Environment is a very, very big factor. And, I wouldn't blame you if you kind of regard it as most diseases as being multifactorial or multigenic. Now we're moving up the chromosome. We went from one gene to multiple genes. Now we're going to actually go into the chromosomes and parts of the chromosome, or this concept of karyotyping. Karyotyping is the study of chromosomes and parts of chromosomes and how they affect, and the diseases that result from changes in these chromosomes. Remember, we're talking about chromosome mutations or damage now, and not necessarily genes. Well, the normal pattern, there's 46 chromosomes. You have 44 autosomes, which are paired, and then 2 sex chromosomes, X and Y. So a normal male would be notated as 46, XY, and a normal female would be 46XX. There are ways of looking at these chromosomes without actually sequencing them and put them into splicing machines. And the classical way, which is really hardly not used anymore because there are so many superior genetic ways of splicing them now, is something that is called G banding, or because the G stood for a Giemsa stain. Giemsa is a stain that's used in pathology for various things. But if you can isolate and blow up chromosomes large enough, at the anaphase stage or metaphase, you could then see pretty good chromosomes. And believe it or not only can you see a chromosome, but with proper good Giemsa banding, you could see 500 different colors or areas, we'll say, within each haploid chromosome. You remember there's a P and a Q arm of a chromosome. The P stands for petite or petit. The Q means the short arm because it's petite. And then the Q arm is the other arm, which is not short. It's the long arm. okay? If you looked at a chromosome, you could group them into how long they are and how they get shorter. So if you take the A group, the B, the C, they are generally longer chromosomes. They have many, many millions of base pairs. Okay, they have many, many millions of genes, perhaps. And then as you get down to the G, you have very short ones, which could be maybe only 1/10 the size, 1/10 the number of base pairs, 1/10 the number of chromosomes. And then if you're a male, you have the X chromosome, which is once again pretty long, and then the Y chromosome, which is pretty small. Believe it or not, there have been some studies to suggest that the reason why more males are conceived and born rather than females, although females generally live longer, is because the difference in the mass of these two chromosomes actually slows down the female a little bit. So could just say the male sperms are perhaps a little more peppier than the female sperms, or the Y. So yeah, I guess you could say that. I don't know if that's true, but I've read a couple of studies. Once again, you go from A to G, from A the longest to G the shortest, and then you have your P and your Q. But within the P and the Q now, the P and the Q arm, you now have regions. And within each region, you have a band. And within each band, you have a subband. So now you could see why perhaps in a single chromosome, you could see hundreds of different stained areas. And this isn't even involving splicing at all. This is simply good microscopy with staining. This is a chromosome. And there's a centromere. So this is shorter, so that would have to be P. And this was longer, so that would have to be Q. And then within the P, you have a couple of regions. And then within the Q, you have a couple of regions. And then within the regions, you have 2 or 3 bands. And then you have subbands within each band. And believe it or not, you could then see that. And if this happens to be an X chromosome, and you have a certain X-linked disease, you can actually pinpoint not only the arm, but the region, but the band, but the subband that the defect is in. Isn't that something? So this is a good mapping of most of the common sex-linked diseases. I don't know if you could see the letters here. I could barely see them myself, but I think we're going to have a better picture of them too. Something that has really kind of revolutionized Giemsa banding to the point where it's not really done anymore is this concept of FISH. And if you want to think of FISH, which is called fluorescent in situ hybridization, as basically the gene probes, you could take small fragments of DNA, perhaps maybe only 10,000 base pairs, for example, and you could see if they match to certain regions of a chromosome. And they're fluorescently colored. So if they match and they go match with the chromosome, then you know that that particular thing is present. Or if they don't match, they know it's not present. So you might think of this as greatly enhancing gene banding, because now you're actually not just staining them, you're actually giving them their little complementary fragments of DNA. FISH is used very commonly in pathology to look at either translocations, which result in many different diseases, especially the hematologic tumors, or whether there's a problem with telomeres, because the telomeres limit the number of times a chromosome can copy. So if it's altered or it doesn't work right, then it could be unlimited cell growth because of unlimited mitoses. And what does that mean? It might be one of the big factors in cancer. In fact, it is. Or you can see if there's been a subtle micro deletion, because now you're looking at 20,000 base pairs. And if you remember, a classical gene has anywhere from usually around 27,000, but it could go up as much as about 20 million base pairs. And a chromosome could have anywhere from 50 to 250 million base pairs. So the A group could have like 250 million base pairs, and the smallest ones can have, just perhaps only 50 million. Here's some examples of fish. In this one, you could see that there has been fluorescent binding, so it shows that there's a triple chromosome 20. So maybe this is a trisomy 20, okay? In this one, you have a deletion, a partial deletion, perhaps a very small deletion in chromosome number 22, and that shows up as a little dot as well, all because of these little fluorescent probes that you're combining with it. I don't have much to say about spectral karyotyping. It's a beautiful, beautiful technique, and I have it as the first slide on our PowerPoints here, only because basically there are ways you could false color the various chromosomes, although some of them will be close in color to the other one, and that's just an electronic trickery. I mean, there's nothing chemically different about these ones, but it's basically what they call a false color computer-generated thing. But don't they look spectacular? As far as I know, I don't think there would be any specific diseases or that could be basically studied very well by spectral karyotyping, other than maybe trisomies or something, or monosomies. So, what is a cytogenetic disorder? Very simple. A cell that has a normal number of chromosomes, whether they're autosomes or sex chromosomes, X or Y, is said to be euploid, with EU meaning normal or good. If it's not normal or good, then it's called aneuploid. In other words, you do not have an exact multiple of 23. Perhaps you have a monosomy, which is a chromosome that is not matched, in which rather than having 46 chromosomes, you have 45. The monosomies of the autosomes are usually lethal, but you could have a monosomy of a sex chromosomes, in other words, a woman with only one X chromosome rather than two, which is not lethal and sometimes very, very livable, although it has its problems. And that's called Turner syndrome. You might remember that. Or you could have trisomies. Many, if not most, of the trisomies or extra chromosomes are also lethal, but there's a handful of them which are not lethal, but autosomes which result in severe problems, of which Down syndrome or trisomy 21 is not only the most common, but it's the most common reason for mental retardation in children. You can have chromosomes, deletions, breakage. Actually, if you take a string, if you put a string in your hand now, take off your shoestring and just look at that string, that's a chromosome. Think of all the crazy things you could do with it. You could tie it in a knot. You could make it into a circle. You could cut part of it out. You could combine it with another part. You could delete it. So these are all the possible cytogenetic disorders. This is not medicine. This is not even science. It's basically mathematics. How many possible disorders could you have with a regular string? Well, in this one, you could have a translocation, which part of one chromosome has crossed over to another part of a chromosome. And that's present in many malignancies and many other diseases too, especially the hematologic malignancies have a whole wide number of translocations. Or you could have an isochromosome, which rather than splitting laterally, let's say, at the centromere, it splits in the other dimension. So the two short arms are basically different from the two long arms. Imagine that blunder, okay? Or you could have a deletion, okay? Or you could have an inversion, a part of the chromosome that's basically switched around. Or you could have a ring chromosome, something that's a circle. These are all possible and seen, and some are related to diseases, and probably the translocations are the ones that are probably present in a wide variety of tumors, especially malignant tumors. So what are some of the chronic diseases involving whole chromosomes, or we'll say mutations at the genome level? Well, you could have an extra autosome, trisomy 21. That's the Down syndrome. The old name for this is the mongoloid or mongoloid babies. And of course, that's very, very politically incorrect. So that was dropped. Probably you'll never hear it. Trisomy of 8 and 9 is possible. There's a syndrome called Pathology, which is trisomy 13. There's a syndrome called Edwards, which is trisomy 18. Most of the other trisomies are lethal. These are ones which are very, very serious. The most significant deletion chromosomal defect is in chromosome 22. And it's in the long arm, which means it's Q. And it's in the region 11. And it's in the band number 2. And if you take that part of the chromosome and you delete it, that results in a very, very serious disorder involving immunity and a whole bunch of other things, and I'm going to talk about it a little bit more. The chromosomal, sex chromosomal diseases are much more common. Turner syndrome is the lack of a complementary X chromosome in a female, and Klinefelter syndrome is an additional X chromosome, and sometimes 2 or 3 additional X chromosomes in a normal XY male. And these, as you would guess, would also result in a male having feminization characteristics, or a female, besides having some masculine characteristics, also other problems. Let's look at the normal classical trisomy 21. You may be given at some point in your life an exam where you're looking at chromosomes, and if you carefully look at how they're grouped, if you see there's a third or extra one at 21, well, you know that's trisomy 21, that's Down syndrome. Or you may have 2 X chromosomes with a Y given you as this karyotyping chart, so you know that that's probably going to be Klinefelter's. Or you may have a single Y with no complementary Y next to it. Okay, so that would be a Turner's syndrome. Let's talk about those a little bit more. In trisomy 21, it's the #1 cause of mental retardation. I don't know if it's in the world, but in the United States at least. You have a trisomy. Okay, most trisomies are present because in the process of anaphase, in which the chromosomes are pulled apart, something goes wrong. Either there's a lag or it doesn't happen, and therefore you now have a cell which has an extra chromosome. Okay, this is also related to maternal age. Okay, and you could also say that anybody that has a trisomy 21 is at risk for things like acute leukemias, certain congenital heart defects like coarctation, GI atresias and narrowing of the gastrointestinal tract. You know, I've always had this little expression here, and some of my students have called me on it. Why would you say they're the most lovable? Well, if you or somebody in your family had one, I think you would agree with my statement here, because we had one in my wife's family as well. Another thing about Down syndrome, and I can't explain it, but I want you to understand it. If you've seen 1 or 2 or 3, if you go onto Google image right now and you just say Down syndrome image, you're going to see like a whole bunch of them. But after you see them in the supermarket, 1 or 2, you're just going to know that it's Down syndrome. There's something very, very classical. Yeah, you could describe the eyes or the nose or the jaw, but forget it. Just use the recognition part of your temporal lobes, and you could say, if you were to see this girl in a supermarket, you would say that's a Down syndrome. Okay? For some reason, they're very easy to remember from facial recognition. Here is a important, probably the most important and well-studied chromosome deletion syndrome. It's chromosome number 22. It's Q, which means it's the long arm. It's 11, which means the region, and then 2 is the band. So if we actually do the Giemsa type labeling of it, you're going to see that in this case, this little segment right here, because you have the Q, and then you have the 11, and then you have the 2, it's just gone. So this is a person with chromosome 22q 11.2 deletion syndrome. Now that's one name for it, and there's probably 10 more other names for this syndrome. It's called DeGeorge's syndrome, which, you know, is nice. It's also called velocardiofacial syndrome because many of the abnormalities in these patients are in the soft tissue and the heart and in the fascia and in the face. It's called congenital thymic aplasia because it results in inherited defects. I'm sorry, in a defect in the T cells, okay? But if you want to just remember, you know, what I think is the best name for it is it's called the Catch syndrome, C-A-T-C-H. Now, I am not a big fan of mnemonics, except for maybe cranial nerves. But if you remember, if you could remember, you don't have to remember it, that the Catch stands for C, cardiac abnormality, especially tetralogy of Fallot. The A stands for abnormal facies. You already know that. The T stands for thymic aplasia. So this is a patient that could have severe immune T cell problems. Cleft palate, because remember we said that the velocardiofacial, the velo is the palate, and hypocalcemia, because in the embryologic development of the parathyroid gland, it overlaps with some of these defects that we see in the face as well. So learn the mnemonic. And yeah, you might also remember that it's a chromosomal. If you want to remember, it's the long arm of 22. That's nice as well. You know the thymus is involved, calcium is involved. And it can be detected by the usual karyotyping in fish, maybe even G-banding. Here are some kids, poster kids for Catch or DeGeorge or velocardial or 22q syndrome. I don't know how I would say them. They're not nearly as distinctive and easy to remember as the patients with Down syndrome, but all of them that I've seen, and you could Google them, you know, Google 100 of them. I just say they all have really cute smiles, okay? Let's move from the autosomes to the sex chromosomes. Let's talk about the 2 main sex chromosome disorders. One in male is called Klinefelter, and the big one in female is called Turner syndrome. Both of them are similar in that they're usually not really suspected or discovered until time of puberty, when the sexual differentiation takes place, secondary sexual characteristics. There may be some degree of retardation in Klinefelters, but if a Klinefelter is only 1X, there's probably not going to be any retardation. If it's 2, 3, I've seen many as 4 or 5X, not personally, but case reports. The more X chromosomes you have, the more likely there's going to be mental retardation. And remember, I don't want to talk about gender today. Gender, this crazy gender politically correct stuff makes me sick, okay? We're talking about sex. That we're not talking about the crazy genders that we're forced to believe now. We're talking about sex. So what's the definition of a male? A male is a person that has at least one Y chromosome. Females are a little bit harder to describe, but I would say if you have 2 chromosomes, X chromosomes, and nothing else, you're a female. If you have only 1 X chromosome, you're a female. If you have an X chromosome, 2 X chromosomes, but the Y, you're probably a male. So remember, very easy. If you have a Y, you're a male. In Klinefelter syndrome, which is an extra X or 2 extra X's or even 3 extra X's, The main problem is hypogonadism, short penises, okay? It's the #1 cause of male infertility. You probably know some people that are Klinefelters, and you'll never suspect it. They don't become retarded unless they start get multiple X's, but most of the time it's just one additional X, which is 82% of the time. Now I have a way of remembering this from its phenotypic expression. They're tall, and most of the reasons why they're tall is that they have long legs. So I don't want to be funny or anything, but they have long legs and short penises. Okay, so they got 2 long legs that should be longer than normal, but the middle leg is a lot shorter than normal. And here are some other secondary characteristics. Because they have additional X, they have some feminizing, things like a feminine pubic hair pattern or some breast development as well. Now let's go to the Turner syndrome, which is a woman that only has 1 X chromosome rather than 2. So she's not aneuploid. She has 45 chromosomes rather than 46. So the proper designation is 45, X. They could have this pattern just in the gonads and not in all of the cells, so mosaics are common. And very often, it's not even the whole X chromosome that's present, but could just be part of it. When you look at all of the features and the classical descriptions of Turner's, the 2 things that are always mentioned near the top of the list is webbing of the neck, okay, and streak ovaries. It means their ovaries don't look like actually, small round structures. They're actually streaked. They're not little nodules. They're just like cords or streaks. They often have edema of the hand dorsum, and they have congenital heart defects as well, of which coarctation is probably the most common and the most feared. And here is a typical Turner syndrome. They also have sometimes little dots, elbow deformities. But if you don't have a great memory, just remember 2 big things: neck webbing and streak ovaries are the classical descriptions. What's a hermaphrodite? Technically speaking, a true hermaphrodite is somebody, Hermes was a god, and he had a sword, and Aphrodite was a goddess, and she had a shield. So you have the sword of Hermes representing the male symbol, and the shield of Aphrodite representing the female symbol. Hermaphrodite would be technically somebody with both, technically both ovaries and testes. These are very, very, very rare. I don't know. I tried to look this up, but I bet you out of the 7 billion people, there can't be more than a handful that are true hermaphrodites. Most likely, 90% of the people that we call both male and female are males with feminine characterization or females with male characteristics. Okay, so remember, even though we talk about Y chromosome, meaning you're a male, and we said that 2 X's, technically speaking, without any Y's, would be a female. There's other kinds of sex also. There's a gonadal sex, the presence or absence of testes or ovaries. And these aren't always necessarily related to the XY scheme as well. Or you could have phenotypic sex, or the presentation of sex, like a penis or clitoris. Or you could have ductal sex. You might remember that in females, the Mullerian duct gives rise to most of the internal female organs, and in males, the Wolffian duct gives rise to many of the male ductal organs. Well, that's another way of looking at sex, okay? We're down near the end. And I want to say a word about a couple of things, is that all of the genetic diseases we've been talking about follow the old, age-long, eternally correct principles of Gregor Mendel, Remember the monk that worked with peas, and he figured this whole thing out way before we knew anything about DNA or genes or anything. So there are certain types of what you would call genetic diseases, which are non-Mendelian inheritance patterns. They don't follow the classical autosomal dominant, autosomal recessive. And one of them is, we talked about the sex-linked disease called the Fragile X, which was the first one discovered. And what that results in is, in the process of the mutation, there is something which causes the nucleotide, in this case it's CGG, to repeat itself many, many, many hundreds, thousands of times, okay? Now, since we've discovered that, there are other types of single gene non-Mendelian diseases, like certain types of ataxias, or another type of muscular dystrophy called myotonic dystrophy. And remember, DNA is not only in the nucleus, it's not only in the chromosomes, it's also in the mitochondria. That is given rise to the theory that in history, maybe mitochondria were parasites which wound up giving oxygen to the cells, so we stayed with them for millions or billions of years. So you can have a non-Mendelian disease. The classical first one that was discovered was an eye problem called hereditary optic neuropathy, which follows problems with DNA in the mitochondria and not in the nuclear chromosome. So, okay. Now here's another type of disease, which for me, I've been trying to figure this out. I know that it has a wide variety of certain diseases. It's called genomic imprinting. And the best way I could explain it is that the gene expresses itself not like it's supposed to express itself, but it expresses itself from the maternal or paternal allele. Okay, isn't that crazy? Well, that's the concept of genomic imprinting. I think it's an infancy stage. I wouldn't be surprised if we have a whole bunch more as time goes on. Maybe there is a bunch more, and I just haven't updated myself. Another thing is the concept of gonadal mosaicism. When we talk about defects in cells, defects in chromosomes, we're assuming that they're in all of the cells, all of the chromosomes, but they could be in only certain cells. And the most common type of mosaicism are the gametes, like either the ovary or the testes, that have the mutated cells, and the rest of the cells of the body don't. So these are all examples of non-Mendelian genetic disorders, and they're all single gene as well. Okay. Everybody who watches TV, and I don't, and I hope you don't either, but everybody thinks that pathology is solving crimes and going to the DNA lab. And, but actually, DNA probes are used in molecular diagnosis for a wide and massively growing number of reasons, especially in the area of cancer. Not only classification of tumors and genetic defects present in tumor cells, but if that particular gene defect would make it more responsible to immunotherapy. So if you have a birth defect, you may not find easily a gene, but we're finding more and more of them, not only postnatal, but prenatal as well. Tumor cells. That's what Jim's lab is all about. He does an awful lot of work in not only classifying tumors with genetics, but telling whether a mutation can result in a certain type of improved immunotherapy. And I have to tell you, the chair that I'm sitting in right now was once occupied by a lawyer who had a renal tumor, and Jim found a certain type of mutation present in that tumor cells, resulting in a type of immunotherapy which that lawyer would have never gotten. Are you ready for the punchline? the lawyer's oncologist had the nerve to use the C word, in other words, cured. And of course, you know that we can identify pathogens by looking at tiny amounts of blood and being completely specific and sensitive for finding pathogen DNA. In the old days, we used to do blood cell typing for donor compatibility or paternity testing. Now the genetics tests are much better. We all have watched CSI. You know, you could probably catch a lot of murderers by looking at the DNA present, which is chiefly mitochondrial DNA, by the way. Here's a concept that was recently made aware of, is that we know that when a malignancy becomes more invasive and with time it becomes metastatic. You can have metastatic tumor cells floating around in the blood or in the lymphatics or in the liver or brain or lungs. But I got news for you. Certain malignancies that have not metastasized can show actually tumor DNA in the blood. How does that grab you? So you can have tumor DNA in the blood in a person with malignancy without actually having metastatic cells. Doesn't that blow your mind? Okay, and I'm sure that would probably make a huge amount of difference, you know, prognostically as well. Finally, here's something that I always kind of laugh at. You know, in the old days, you know, in the Stone Age, when I was a pathologist, we would look at a slide, and a lot of the slides you're seeing in this course and in the laboratory course are H&E, hematoxylin, and you look at structures, you diagnose tumors, you diagnose diseases, okay. And then probably in the last 30 years or so, there has been a big wave to look for antigens, immunoantigens with a technique called IHC or immunohistochemistry to define a wide variety of antigens, not only to tell, you know, where that tumor cell came from, but also massively important for prognosis of the patient. Now, many people have told me in the last 10 years or so that genetics is going to take away the microscope and it's going to take away the IHC, because not only now you are not looking at antigens, but you're looking at the genes that made those antigens. So wouldn't that be interesting if the pathologist 20 years from now doesn't have a microscope, but he's looking at these little dots all day, saying, oh yeah, that gene is mutated, that gene is should be there, that gene is overexpressed. Isn't that something? Because when you think about it, that's really the basis of all diseases, isn't it? So we can move technically from histology to immunochemistry to gene microarrays or sequencing. But guess what? Today on planet Earth, still the main diagnostic technique, probably over 90% of the time, maybe 95, is regular old H&E. So there you go. Let's call it a day. Thank you for being here. I feel like this webinar went very good. I'm very glad I had the energy and inspiration to talk about this very, very complex topic. I hope you had fun, and I'm going to play right now for you the very, very last Phil Collins Genesis Genetics song.